Hello guys, how are you? Very nice to have you here today. My name is Belinda and I'll be presenting today's session. Okay, so we will be going back to term 1, revising the topics that were done in term 1. Okay, with the other grades, what I do was I revise the most challenging topics. But with grade 12, it's important that we learn each and every topic and we make sure that we understand each and every topic. So I will be going back to term 1. Today we're going to be looking at protein synthesis. Okay, so if you have done it before, this can be a good revision opportunity for you. Please attend. Okay, some of you have done it already, but it's a revision. Okay, the more you do it, the more it's gonna stay in your head, so you're not wasting any time. Okay, so before I start, please make sure your audio is on and your speaker volume is turned up. Please note that you are muted when joining the session. So if you have any questions, please use the question box to send me questions. Also, you can download the handouts from the handout section. You are encouraged to ask questions, okay? But please do not ask any irrelevant or inappropriate questions. Only ask questions that are relevant to this content that we're discussing today. If we did not get to your question, please send an email to academics@impact.co.za, okay? We will discuss any questions that you guys send after the session if we still have time. Okay, for past sessions or class videos, you can visit www.impact.co.za, COVID-19 online schooling. Yeah, you will find all the past recordings there, as well as this recording today. Okay, so we will be doing term 1 revision as well as term 2 revision in the next couple of webinars that we're gonna have. Okay, they're still gonna send you a schedule with regards to that. Okay, we are not going to be writing a June exam, and I'm sure they've communicated that to you, but you will be writing a final exam on these topics. So writing practice June exam paper is good practice towards your final examination. Okay, so in term 1, you've worked through DNA replication and photosynthesis, which is what we're going to be looking at today, meiosis, genetics, as well as reproduction in invertebrates and humans. Okay, these are the topics that you did in term 1. So before we start, I'd like you guys to please, on the question box, send me a list of questions, or even if not questions, or the topics that you found the most challenging or difficult to answer in your first test. So if you feel like you didn't do well in your first test, what topics did you find challenging? Okay, what were you not able to answer correctly? Can you please send me those problem topics on the question box, and then we will be focusing on them with binoculars in the next sessions when we're doing revision for term 1 and 2. Which topics were problem topics for you? Okay, you can also go deep into a topic and find a specific section that you didn't understand, or a term. Okay, if non-disjunction was there and you couldn't figure out what it was, or if you were confused with crossing over or whatever in meiosis, you can type it now and tell me, okay, I didn't quite get this. Okay, I know the menstrual cycle has proven to be a challenging question for you guys, especially the graph. If you found it challenging, please write it in the question box, okay? Leave no stone unturned, right? Whatever that you would like for us to discuss, okay? We will discuss it in the next session. I'll include it in my slides in the next question. Okay, I'm just gonna give you guys a few minutes, 1 or 2 minutes to write it down. And yeah, while we wait for everyone to get settled. Okay, from term 1, what did you find? What did you struggle with? Which questions did you struggle to answer? Okay. Another thing, when answering questions in a test, sometimes we get them wrong because we didn't understand or we didn't read the question carefully. So whenever you get a question, please take some time to read the question thoroughly and don't be quick to answer. Understand exactly what the question is asking you before you attempt to answer it. Okay, it's very important because sometimes a question is really easy, but because you didn't understand what was asking you, you will answer it incorrectly. Okay, use highlighters, use colors, whatever you can that will make you pay attention to the question and not answer it incorrectly. Okay, so today we'll be looking at protein synthesis and DNA replication. Okay, let's start. Okay, so protein synthesis as well as DNA replication takes place inside the cell. So we'll be digging deep inside the cell today. Okay, we will also break it down to in which organelle in the cell will we be working in. Okay, so we're gonna break that down and we're gonna further go into that organelle and see what inside that organelle is responsible for DNA replication. Okay, so we'll be going deep into the cell today, right? Okay, so the nucleus is the organelle in the cell that we are most interested in today. Okay, the nucleus is the most prominent organelle in all eukaryotic cells. By eukaryotic, they simply mean cells that have a nucleus. Okay, so it is found in a eukaryotic cell, cells that have a nucleus. Okay. Okay. What the nucleus does is it stores all the genetic information. It stores all the genetic information in the genes of the chromosomes. So inside the nucleus, you will find chromosomes, these X-like structures. You will find them. This is where genetic information is stored. It is stored inside the genes in the chromosome. Okay, we're gonna look at a magnified view of the chromosome and where exactly the genes are found. Okay, in the chromosome. Okay. The nucleus is the CEO of the cell and it directs all the functions of life and in addition prepares the cells for maturity and replication. Okay, so it controls all the functions for life and prepares the cell for replication, which we're going to be looking at now. Okay. Inside the nucleus, we have X-like structures called chromosomes. Okay. A chromosome is an entire chain of DNA wrapped around proteins called histones. A chromosome is an entire chain of DNA wrapped around proteins called histones. So we know that this is how a DNA structure looks like. This is a long chain of DNA. You can see it with that double helix. So this DNA wraps itself around this stones called, this proteins called histones, and wraps itself around until it forms an X-like structure called a chromosome. Within this chromosome, or within this DNA, a section of a DNA contain a gene. Okay, we will look at that next. Some sections of the DNA contains genes which code, or which has genetic information that code for our proteins, that code for our hereditary features. Okay, humans have 23 pairs of chromosome in each cell. So in each cell of a human, you will get 46 chromosomes, or 23 pairs of chromosomes. So when they say 46, they are counting the sister chromatids individually. So this will be chromosome number 1, number 2, number 3, number 4, number 5. So you can either say 46 chromosomes altogether, or 23 pairs of chromosomes. So inside a living cell, you will find 23 X-like structures in simple terms, if I was to put it in simple terms. 23 X-like structures. Okay. These chromosomes are further broken down into smaller pieces of code called genes. Okay, the 23 chromosomes together have about 70,000 genes. So the 23 genes, 23 chromosomes inside each cell in your body contains about 70,000 genes, and each gene has its own function. Each gene has its own function. This is just for enrichment. They will not ask you in the exam. You're not going to be asked how many genes are found in the cells. Okay, this is just for enrichment. It will not be examined. Okay. Please send questions as we go. Also post the questions that you found challenging in your, or that you got wrong, that you don't understand how or why you got them wrong in your examination, and I'll gladly clarify that for you. Okay, so please do that as we go. You can also, if you are doing impact, the impact curriculum, you can also just specify the questions. Say maybe question 2.2.2 or 2.2.1 or whatever it is. I have the question paper, so I will have a look for you. Okay. So we said on the gene, on the DNA, we find a section of a DNA is called a gene. Okay, each gene has genetic information. Okay, so this is a section of a DNA and it is called a gene, which contains genetic information that codes for a specific protein. So each gene has a code or genetic information that codes for a specific protein. This protein will give your body instructions on how it should look like, or how it should perform certain functions. Okay, for example, this was a genetic code or a gene that had genetic information for coding a protein that will make your thumb like that. Okay, not all of us have that kind of thumb. Okay, but this person had a genetic or a gene that was able to code for that kind of thumb. Okay, being able to bend their thumb like that. Okay, so section of DNA I called genes. Okay, so let's look at an overview of what happens during DNA replication. Okay, we're gonna look at what happens before, what happens during, and what happens after DNA replication. Before DNA replication happens, okay, remember mitosis from grade 10. We won't be going through mitosis, but it is involved somehow there. Okay, after mitosis is that cell division whereby a cell duplicates itself, okay, for growth, for repair, and for replacement of dead cells. After mitosis, the cells grow, okay. While some mature and specialize, some will continue to DNA replication, and some will divide again, okay. Some of the cells will mature and specialize, and some will replicate their DNA. Okay, during DNA replication, what happens is each of the 46 DNA strands make a copy of itself, so that there are now 2 strands of DNA. Remember we said a human cell has 46 chromosomes. So each of the 46 DNA strands make a copy of itself, so that there are now 2 strands of DNA. So that's what DNA replication is. And then what happens after DNA replication? The cell will continue to grow, synthesize proteins, undergo other metabolic activities, and begin to prepare for mitosis. And then the cycle happens over and over again. All right. So let's just look at a brief history of where this began. So it began in 1856 with a natural science teacher who was working in his garden with pea plants. His name was Gregor Mendel, also known as the father of genetics. Okay, so during his experiments, he discovered that parents have a specific elementine that can be passed on to offspring. So that's what he discovered. He discovered that parents have a certain elementine that they pass on to their offspring. But what Mendel did not know was that that elementine that he was talking about, or elementin that he was talking about, was found on chromosomes. He did not know that. And it was later, with the help of research from other scientists, it was later, that elementine was later renamed to genes. Okay, so he made all this possible. Without him, we wouldn't even be able to study this DNA replication at all. Okay, sorry, let me go back. Okay, so we're gonna look at the DNA structure first. Okay, DNA consists of 2 molecules that are arranged into a leather-like structure called a double helix. It's very important. A DNA structure is a double helix. It winds together to form a double helix. Okay, a molecule of DNA is made up of millions of tiny subunits called nucleotides. So nucleotides are the building blocks of DNA. Just like atoms are building blocks of matter, and cells are building blocks of life, and amino acids are building blocks of proteins, nucleotides are building blocks of DNA. Okay, so each nucleotide, the building blocks of DNA, consists of a phosphate group, a pentose sugar, and a nitrogenous base. Pentose just means that it's a sugar that contains 5 carbons. Okay, so each nucleotide consists of a phosphate group, a pentose sugar, and a nitrogenous base. Okay, this is what a nucleotide looks like. Each nucleotide has a nitrogenous base. It's got a pentose sugar. You can see it 1, 2, 3, 4, 5. Remember, pent means 5, and it's got a phosphate. Okay, so now we have 4 different types of nitrogenous bases found in DNA. The first one is adenine, and then we have cytosine, labeled C. We have thymine, labeled with a T, and we have guanine, labeled with a G. Okay, so those are the 4 nitrogenous bases that are found in nucleotides. Okay, these are called complementary base pairs because they are always paired together. The adenine always pairs with thymine, and guanine always pairs with cytosine at GC. Okay, so these are called complementary base pairs, as you can see that they are perfect fits for each other. Okay, adenine is always paired with thymine, and guanine always paired with cytosine. Okay, each base will only bond with one other specific base. Adenine only bonds with thymine. Guanine only bonds with cytosine. Okay, these are called a base pair. This is another base pair. Okay, complementary base pairs. Okay, remember to please send through your questions. Because of this complementary base pairing, the order of the bases in one strand determines the order of the base in the other strand. So if you've got a cytosine here, we're going to have a guanine there, we're going to have a thymine here, we're going to have an adenine there. They're always the one strand will determine what the other strand is going to be. Okay. Can you guys, in your question box, tell me what the other strand of the DNA is going to be? What will this side look like? Okay, just give me the letters in that order. What will the other strand be? Just give me the letters in your question box. Remember, A with T, G with C. So can you tell me what the bases on your other strand are going to be in order, looking at what the base for the other strand is? Can you type it in your question box? What will the other strand of the DNA be based on this one? A will pair with T. G pairs with C. Okay, thank you for your answers. Let's check if you guys were correct. It seems like most of you are correct. Okay, well done. So let's just check. Yes, so it's T, G, A, G, T, C, A. All of you got it correct. Well done, you guys. Okay, so this will be bonded together by weak hydrogen bonds. Okay, and we have our double strand. We have our double strand of DNA. Okay, so to correct the genetic code found in DNA, we need to look at the sequence of the bases. The bases are arranged in triplets called codons. Okay, so each triplet is called a codon. So in order for us to crack what the code is for, or what for what protein this is coding for, we need to look at the sequence of the bases. Okay, T, C, in that order. G will be paired with C, A will be paired with T. Okay, so this is a sequence for some sort of protein. Okay, normally in your examination they will give you the sequence, and then they will give you the type of protein, and then they will ask you questions based on that. So you don't have to memorize those protein in your exam. They will give it to you in a table format. Okay, we've looked in the previous sections that a gene is a section of DNA that codes for a protein. Okay, each protein has its own function. It can be a protein that tells us what the color of your eyes can be, or it can be a protein that functions in your muscles, or it can be for anything. So each gene is found in some part of the DNA. Each unique gene has a unique sequence of bases. This unique sequence of bases will code for the production of a unique protein. It is these proteins and combination of proteins that give us a unique phenotype. Remember what phenotype is? Your appearance. So these proteins and give us, or tell us, or they decide what we should look like. If whether our hair should be curly or straight, if our eyes should be brown or blue. So these proteins are responsible for that. What is the role of DNA? This could be an exam question as well. DNA is important for inheritance, coding for proteins, and genetic instructions for life. So this makes DNA important. Okay, genes make up, this is just for enrichment, it won't be asked in your exam. Genes make up only 2% of the human genome. The rest of the DNA is made up of what we call non-coding regions, some of which regulate how your chromosome should look like, and also how many proteins should be produced and when they should be produced. Okay, so the function of that is not known. It does play a specific role, but it's not known. And that DNA is called junk DNA. Okay. DNA replication. Okay, let's look at this. Steps of DNA replication. Okay. When a cell is ready to divide, each divide by mitosis. Okay. Each DNA molecule duplicates itself. Both strands of DNA in the parent cell act as a template for the formation of the new complementary strands. Okay, so this is what happens. Step number one, okay, so we have a normal DNA here, and it's a double strand, and the base pairs are bonded to each other. Okay, step number one is that the DNA helix will unwind. There it is, it unwinds. Okay, so it goes from being winded to unwinded. So that's step number one. Step number two, the hydrogen bonds between the bases will break, allowing the two strands to unzip. So remember, these two nitrogenous bases are bonded together with hydrogen bonds, weak hydrogen bonds. So they will start to break, and then the two strands will unzip. Okay, those nitrogenous bases will then become what we call free nucleotides. They will be free to bond with others. Okay, they are single and ready to mingle now. Okay, this exposes the bases on the template strands. So they are now exposed, so they are ready to bond with other ones that are free flowing in the nucleus. As a result, free nucleotides are brought to pair in with the exposed complementary templates. Okay, so this was exposed and this one was exposed. So free nucleotides from the nucleus will see an opportunity to bond with the exposed one. Okay, as a result, we're gonna have two new DNA that's being formed. Remember, adenine always pairs with thymine and guanine pairs with cytosine. Okay, so you must remember that when this happens. Okay, then the DNA strands will wind back together. They will wrap around a histone and a new chromosome will be formed. An X-like structure will be formed. Okay, I hope this has cleared the misunderstanding or misconception. If you want me to repeat it, please can you write it in the question box if you did not understand. Or can I ask you guys to raise your hands if, let me put this down. Can I ask you guys to raise your hands if you understand the steps in DNA replication? Please raise your hand if you understand the steps in DNA replication. If you don't understand, please type it in the question box for me to repeat. Okay, please type it in the question box if you'd like for me to repeat this. Okay, somebody wants me to repeat, I shall do so. Okay, so we're gonna start with a normal DNA, which is winded together into a double helix. Step number one, this double helix will unwind, this will unwind. Okay, the double helix unwinds. Step number two, the hydrogen bonds that were keeping this nitrogenous bases together will break. After breaking, the strands will then unzip. So first step, they unwind, and then the hydrogen bases, hydrogen bonds that were keeping the bases together will break, making the strands to unwind. After unwinding, this bases will be exposed. Okay, they will be exposed, and free nucleotides from the nucleus will want to bond with them because they are now exposed. So free nucleotides will want to come and bond with them. So when adenine sees a thymine base, it's going to run to it and bond with it. And when a cytosine sees a guanine base, it's going to run to it and bond with it until we have two strands of DNA. Okay, and then they will wind together. The long chain will wind or wrap itself around histones, and a chromosome will be formed. Okay, I hope. Now we're going to look at protein synthesis. Still have a bit of time. Okay, this is an interesting fact. The longest English word takes about 3.5 hours to pronounce and contains that amount of letters. Let me just open my slide. The longest English word takes about 3.5 and it contains about 189,819 letters. And this is a giant protein. It is the name of a giant protein. And for convenience, it was given the name titin. Okay. This is the protein that allows your muscles to be elastic. This is its name. All these words that you are seeing here is the name of that protein, and it's not even complete. Okay. It also has the longest IUPAC name, and it takes about 3.5 hours to read. Okay, you can go and find Google videos of people who tried to read this. It took them about 3.5 hours to finish. I don't know if you have 3.5 hours in a day to listen to somebody trying to pronounce that word, but it was given that name, titin. So instead of calling it that, you can just simply call it titin. Okay, this is produced during protein synthesis. Okay, isn't that amazing? It's produced. This protein is produced during protein synthesis. Okay. So let's look at the central dogma of cell biology. Okay. So DNA replicates. Okay. Then from DNA, transcription takes place, which is the process of formation of mRNA, which translates to form protein. Okay, this is called the central dogma of cell biology. DNA replicates, and then from there, the DNA through transcription forms mRNA, which through translation forms protein. Okay, so we're going to be looking at that now. So these are the steps of protein synthesis. The DNA double helix unwinds but partially. Okay, and one strand is used to make one of the 3 types of RNA necessary for protein synthesis. Okay, so we have a template strand, a double helix of a DNA strand. So during protein synthesis, it's going to unwind partially. Okay, it's going to unwind partially. And as it unwinds, the one strand is used to make one of the 3 types of RNA necessary for protein synthesis. Okay. The mRNA is also known as the messenger RNA. Okay, this mRNA is produced from a DNA template. Okay, do you see that DNA? So it unwinded over there, and from this an mRNA was formed. There is the mRNA. But let's see how the mRNA is different. As you can see, thymine is not found in the mRNA. There is no T at all. So in an mRNA, thymine is replaced with another base called uracil. So this DNA partially unwinded, and then an mRNA was formed or from it. In that, the thymine was replaced with uracil. Okay. Then tRNA will take the mRNA into the ribosomes. Remember from grade 10, we learned that ribosomes are responsible for protein synthesis. So the tRNA, also called as transfer RNA, will take the messenger RNA to the ribosomes. This is called the messenger RNA because it's got information from the DNA. It's got genetic information from the DNA. So it's a messenger. This transports it to its site. So the transfer RNA is transporting it to the site where protein is going to be manufactured. Okay. Then the rRNA, the ribosomal RNA, will help in linking the amino acids together to form proteins. Okay, so this is the steps. A section of the DNA will unwind. In this case, this is the section that unwinded. From there, this mRNA was formed. This mRNA was formed. Okay, and then the tRNA will transfer, or it will transfer the mRNA to the ribosomes. The ribosomes are going to read off the genetic. They can read. They are the readers inside the nucleus. They are the smart ones. They're going to read off the code that the messenger RNA contains. Okay, and they're going to start manufacturing that protein. Okay. After the mRNA breaks and is taken to the ribosomes, the DNA will wind back again, and whenever proteins need to be manufactured again, a section will unwind, an mRNA will be formed, the transfer RNA will take it to the ribosomes, where they will read the code and start manufacturing the protein. The ribosomal RNA will link the amino acids together to form protein synthesis. Okay. If you'd like me to repeat this, please type it in the question box. I'll be happy to do so. If you want me to repeat this, can you please type it in the question box? Okay. Okay, I will do that. Okay, so this is how proteins are formed. Let me get my pen. This is how protein is formed. Okay, there's my pen. Okay, this is how protein are formed. Okay, it starts off with the DNA, normal DNA. So first, what's going to happen is a section of the DNA will unwind. In this case, this is the section of the DNA that unwinded. Okay, from that section, one of the DNA templates will be used to produce the mRNA. But instead of thymine pairing with adenine, a new base is introduced called uracil. Okay, all these are found in the free nucleotides that are in the cytoplasm. Okay, so a new base will be introduced. So after the DNA unwinds, one of the exposed strands will pair with the mRNA, or it will produce the mRNA, okay, which will be taken into the ribosomes found in the cytoplasm. So they will be transported out of the nucleus into the cytoplasm, where the ribosomes can read off the information that they have, and from that proteins will be manufactured. Okay. This process of creating mRNA, or creating a section of RNA from the DNA, is called transcription. So it's the process of creating RNA from DNA. Okay, and the process that takes place in the cytoplasm, in the ribosomes found in that cytoplasm, is called translation. So translation is the process whereby the actual proteins are manufactured. Okay, so the process of creating an mRNA strand from DNA is called transcription, and then the process of creating the actual protein inside the cytoplasm is called translation. Right? And as you can see, RNA is different from DNA because it's a single strand. Do you see that? Unlike the DNA, RNA is a single strand. Okay, it's a single strand. Okay, this is a simplified structure of what happens. Okay, of translation. The ribosomes will start reading the RNA to find out what protein it needs to produce. Okay, so translation, translation is that takes place in the ribosomes. The message contained in the triplet codons of the mRNA specifies a particular amino acid to the ribosome. So the messenger RNA will have information on what protein the ribosome must produce. Okay, then the amino acids are linked to together by the ribosomal RNA and peptide bonds, and then a protein will be formed. Okay, these are not going to be examined, but these are the functions of some of these proteins. Some of these proteins are antibodies that protect us from bacterial infections. Some of them are enzymes that help our bodies with metabolic activities. Some of them are signaling compounds, okay, that are essential for cell-to-cell communication. Some of them provide strength and structure to the cells in our bodies. This will not be examined. It's just for enrichment. Okay, this brings us to the end of our session. If you've got questions, please use this time to type them in the question box. Unless you want me to unmute you so you can ask the questions. If you want me to unmute you so you can ask questions, please type it in the question box. Otherwise, please type your questions on the actual question box and I'll answer it. Otherwise, if you are fine, you are welcome to leave the session and go and redo the questions in your test on this section on DNA replication and mark it again to see if you now understand. So go and redo the DNA replication and protein synthesis part of your paper, as well as try out some past papers as well, just this section. Okay, you're welcome to leave if you don't have any questions. If you'd like to ask a question live, I can unmute you. Maybe something was not clear to you. I can unmute you and you can ask me or type it on the question box. Please, guys. Okay, since there's no questions, thank you guys for joining me. I will, oh, here's a question. paper 2. This is found in paper 2 of your examination. Okay, any other questions? Ask away. Nothing? Any more questions, you guys? mRNA carries the code to make protein to synthesize proteins. Okay, so somebody asked which type of RNA carries the code to make protein. The mRNA does that. The one that is manufactured from the DNA contains the code. The one that is made from DNA contains the code. So this one, this one, the mRNA contains the code. The messenger RNA contains the code. The closing date for the term 2 tasks is August, if I'm not mistaken. 15th of August or somewhere there. This is the closing date for all your tasks, if I'm not mistaken. How did you guys find your task 4 on genetics? Do you have any questions? Okay, anyways, it's almost time up. If you've got any more questions, please send them to academics@impact.co.za and we shall look at them. Enjoy the rest of your day, stay safe and stay healthy. Bye.